And last but not least, we have James Fawcett. Please, James.
So I want to talk at a sort of cellular level about a problem that the other three speakers have referred to in different ways. That's the problem of integrating transplanted cells into the host nervous system. So there's a tendency, when you put cells into the nervous system, for them to transplant as a lump and to stay as a lump, and not to migrate out or integrate with the host tissue. And transplanting a lump into your nervous system is unlikely to be particularly beneficial. So what you would like to do is for the transplanted cells to migrate out and integrate into the host nervous system, where they can influence things like scar formation, vascularization, axon growth, and so on. So this is a generic problem of transplantation. It doesn't matter if you've transplanted bone marrow stem cells, ordinary stem cells, neural crest cells, or what I'm going to talk about mostly today, Schwann cells. So there's a particular issue with Schwann cells, which I'll explain to you. So Schwann cells potentially are of enormous use as a bridge across a spinal cord injury. So here's an old experiment done by the Aguirre lab many years ago, in which a spinal cord here has received a Schwann cell graft, in this case a peripheral nerve, which acts as a bridge for regenerating nerve fibers. And what you would like is for those regenerating axons to continue on and integrate with the host nervous system. The problem, as I'll show you, is that the Schwann cells at the end of this graft do not integrate with the host nervous system. They form a boundary, and the axons can't cross that boundary to re-enter the host nervous system and make connections. So there's a host integration problem. So here's a picture of the sorts of things that happen. This is an in vitro picture of a confrontation culture with Schwann cells in red on the bottom, astrocytes in green on the top, and you can see basically these cells don't like one another. They separate out into discrete areas, and there's a sort of warfare going on at the interface here. You see this also where you transplant Schwann cells in vivo. This green lump here is transplanting Schwann cells, and you can see the amount of integration and migration into host tissue is extraordinarily little. There's a bit of migration of Schwann cells out of this sort of graft, but it's pretty much all down blood vessels, where they're migrating down the blood vessel extracellular matrix. And these cells, again in vivo, are walled off by astrocytes, very much in the way you see in this in vitro model. So what is it about Schwann cells and astrocytes that prevents the two integrating together? So the issue here is, again, comes down to regeneration and repair. You read a lot about protein sulfate proteoglycans, anti-no-go, and things as general inhibitors of axon growth. So you might call that surround inhibition. What we're talking about here is an interface between Schwann cells and astrocytes, which forms a barrier through which axons are simply not going to cross. We see an example of this in a beautiful experiment published by John Goulet some time ago, in which you see axons approaching the end of a Schwann cell graft, a peripheral nerve graft, just as I showed you earlier. And you can see this axon's just done a U-turn. There's no way it's going to cross that boundary from permissive Schwann cells into less permissive astrocytes. And this boundary is marked by a dotted line, but it's a strong boundary. To some extent, in this experiment, they were able to break down this boundary and allow axons to cross by treating with chondroitinase, which Mike also talked about. So I'm going to talk at a cellular level about the interaction between these two cells and how you might allow them to integrate better. And we're going to be dealing with, particularly with two types of assay, what we call the broken coverslip assay, in which in this case Schwann cells have been put on a piece of glass, which has been turned cell side downwards onto a test surface, which in this case is astrocytes, but could be a substrate. And we're going to be looking at confrontation cultures, in which you start with a whole lot of Schwann cells one end, a whole lot of astrocytes the other, and they bang into one another as they migrate towards the middle. So, first of all, I'm going to talk about a candidate molecule produced by astrocytes, agricam. Now, we've been looking at this molecule particularly because it's one of the major glycan-bearing chondroitin sulfate proteoglycans that you see in the injured nervous system. So like all proteoglycans, there's a protein core down the middle here, but all these things sticking out like a bottle brush are the inhibitory sugar sulfated glycan chains, which are responsible for inhibition. And aggrecan is a particularly heavily glycinated chondroitin sulfate protein glycan. Here's another picture of this bottle brush. So is this a reasonable candidate for inhibiting Schwann cell migration into astrocytes and into spinal cord tissue? Well, first of all, is it produced by astrocytes? Yes, it is, both in vitro and in vivo, and at sites of Schwann cell injection, it's also upregulated at the interfaces. Is aggrecan inhibition to Schwann cell migration? The answer is yes, very. These are Schwann cells migrating out from a broken cover slip on laminin with aggrecan added, and here's aggrecan treated with chondroitinase to take away the glycans, and you can see this removes most of the inhibitory activity. So this is glycan-related inhibition due to aggrecan. Is aggrecan important in blocking Schwann cell migration on astrocytes? Well, we can investigate that by simply knocking it down, and when we do that, we do see improved migration of Schwann cells across an astrocyte surface. And finally, can we do anything about this in a slightly more cellular method? Well, here's some Schwann cells that have been transfected to secrete chondroitinase, and you can see that they're digesting the chondroitin sulfate proteoglycans on the astrocyte surface. This is a stub antibody staining, and the green represents digestion. And again, the transfected cells migrate much better than control cells. If you do this in a confrontation assay, again, here's a control confrontation assay. Here's Schwann cells treated with chondroitin sulfate proteoglycan, and you can see they're integrating much better. Sorry, with chondroitinase, you can see they're integrating much better with the astrocytes. Now, how does this work? What we think, I'll show you some evidence in a moment, is the aggrecan works on a receptor yet to be identified, and it affects migration through an effect on integrins. Why do we think that? Well, if you want to affect integrin-related migration, you're probably going to affect integrin activation. So this is an integrin that will bind to a ligand and promote migration and signal through focal adhesion kinase. This is an inactivated integrin that has been affected through tailin and kindlin at the end of the beta subunit.